tell us about Genescape?
So, Genescapes is a project looking into phytoremediation, which is the plant's ability to accumulate heavy metals. So, in this project, we're looking at silver from photographic fixer and looking to upregulate the capability of Mentha piperita, which is peppermint. And by introducing a gene from another species, which is classed as a hyperaccumulator, which are very proficient in extracting heavy metals from the soil, we hope to increase peppermint's capabilities in doing so.
What were the successes?
So the main successes has been artistic output. The scientific side has been challenging, but looking at photography solutions through the microscope and looking at those substances when they're evaporated, being able to visualise contamination within the soils and create composite images of between 50 and 70 photographs stitched together. So these are quite impactful and successful in generating discussions around the topic. Another development has been looking into digital technology and software programs like Touch Designer to create a digital immersive environment for the Science Festival next year.
I would also just add to that some of the successes from a scientific point of view has been introducing concepts of science to someone that only wouldn't have that knowledge. And so from my point of view, that's really interesting. And then also being able to share that sort of joint knowledge from an artist, from a scientist with other lab users, I think is a huge success because there are now more people interested about bringing those two disciplines together.
What were the challenges?
The biggest challenge has been inserting the gene. We've been working on it for over 6 months now. And we've not had success. We're very much closer, as close as we can be to having success. But yeah, the methods have taken a lot longer than was originally anticipated and researched. It's quite novel, this. There's not a lot of studies on silver in particular. So really we're learning through doing rather than through reading. And there's very few existing studies that we can refer to for this project. And I think the sort of strictness of scientific rigour, is very disciplined, and perhaps my artistic practice is not so much. Have a bit more freedom within that. So balancing those two things, I think, has been the biggest challenge.
Yeah, and also just the complexity of what we're doing, and like Scott's really mentioned, there isn't much reading that we can do around what we're trying to do specifically. There are articles and research papers out there about a similar concept, but not for what exactly we're doing. So we're kind of not going in blind, we're going in with some knowledge, but it's completely unique to the specific thing that we're trying to do. And so that's quite a challenge, is that you have this knowledge, you think, oh, that might work, and then it doesn't. Back to the drawing board, troubleshooting it. And also a lot of these processes that we're doing are not instant gratification processes. They take a couple of hours on a day, and then to get the end product, it's 3 or 4 days later, which is an entire week. Didn't work, go back to the drawing board next week, same thing, repeat. It gets exhausting.
Ethical consideration for genetic engineering?
So it was something I was very conscious of before the project started. Within the lab setting, we've got kind of strict protocols which Keira designed at the outset. So everything we do in the lab is very controlled. It's safety level 1.
Yeah.
So there's very little risk of anything leaving the lab, or even if it did, it would have a huge impact. I think if this project is successful, releasing transgenic plants into a post-industrial site, for example, the conversation becomes a lot more nuanced. and we need to think about which organisms are eating these plants and so on and so forth. But within the lab, it's very, very controlled and poses little risk. I think also because we're using it to try and remediate, as a solution to human-induced contamination, people are quite supportive of the project.
What did you learn?
So, I didn't know anything about genetic coding, really. The first fascinating insight was the restriction enzymes being able to recognise palindromes, and that's how it's cut. And then running the gels to determine if the gene restriction had worked. And then doing ligations and then trying to troubleshoot why they haven't worked. So, in the troubleshooting, I think actually through things that haven't worked, I've learned more than had all these processes worked first time. I probably would have just done what was written in the protocols, and I wouldn't have had any reason to look any further.
Yeah, I often think that not mistakes, but like the things that don't work out is how you understand what certain things are doing. Reading a protocol means adding liquid A to liquid B, and if you don't see it not working, then you're kind of taking the research or the protocol at surface and face value, and you're just assuming that what it's telling you is correct. With it going wrong, you're kind of understanding all the steps involved and ways that you maybe try, as I mentioned earlier, about like research papers not having specific information about what we were trying to do. So by things going wrong, that's kind of sometimes not in the best way possible, but it's kind of directing you in a way that maybe is helping and figure it out.
How do you see the project evolving in the future?
So I'd like it to look at other plant species and their capabilities to extract other metals, particularly used in arts practices, mainly photography and printmaking. The idea of this came about. I was making plant-based developers for developing black and white negative film, and that's the reason peppermint's been used. It's high in phenols, which is the main developing agent in photographic solutions. So the idea is that I can grow what I need to develop the film and then feed any contaminants from that process back into the plants and create this closed loop artist ecosystem within my studio. So hopefully other practitioners in printmaking, for example, could do the same with other metals that they're potentially throwing down the sink.
How does your background influence your approach to the Genescapes project?
Yeah, so my background's in engineering. So, I've got a kind of intrinsic curiosity as to how things work. I was like that as a kid. I used to take things apart. Sometimes new presents and unboxing day, they would be taken apart to see how they work. I think that curiosity has been quite helpful in fault finding and troubleshooting on this project. And even when I work in the landscape, I'm also very curious into sort of biological systems. And I think the more you understand the science behind something, the more informed that project can be. I think art's very good at speculative fiction, but it hasn't always got the scientific weight or the research behind it. And it's kind of what I was hoping to build with this project, was something that had a bit more guts in terms of the context.
From my perspective, like with a very academic, scientific background, like my sort of initial approach to the project was, also as the manager of ASCUS, like I've got like health and safety in the forefront of my brain. So starting off the project was the risk assessments, especially working with genetically modified organisms, is that's sort of like a heightened level of risk. Not that can't be controlled, but it was just like an extra layer of thinking I had to do, research I had to do. My background's in microbiology, so I'm less experienced with plants and like the different types of risks that are involved in plants, which I've learned comes from aphids as we learned through the project, and other kind of pests, and there's just a whole lot of different things. So I was quite not concerned, but just like the priority for me at the beginning of the project was trying to make sure that the lab remained a safe environment. But then also like I'm quite meticulous, and so I spent a lot of time designing the protocols like step by step, which at the end of the day ended up just getting abolished.
How do you measure the success of an interdisciplinary project like Genescapes?
So the main thing is to generate interest beyond art and science. We're lucky enough to have an output for the Science Festival next year. So exposing the research and the work to a non-arts or non-science audience is great, but also a science audience coming into the exhibition and vice versa is also really interesting. If I can generate intrigue in the audience, then it opens up a conversation on their terms, which is hugely valuable as opposed to reading an academic paper, for example. People tend to shut off, or likewise, if we're having a conversation, people feel like they're perhaps getting preached at. So yeah, to get their interest in the project is fundamentally what I want to create, and then hopefully that creates a further discussion within the photography community, within contemporary art, and within science.
Yeah, and just to add to that as well, the lab is a community space, so we've got a variety of different people from multiple different backgrounds that come into the space, and when they see plants growing in a fume hood and they kind of thinking, oh, that's interesting. Like they ask me about the project and I will tell them about the project, and you can kind of see some people confused, some people like, that sounds really interesting. And so that I feel is a measure of success as well, because you're getting people that really wouldn't even think twice about this type of thing to see it, think about it, ask questions about it, maybe go home and read about phytoremediation, or even if it just makes them consider their waste in a slightly different way.
Exactly.
That's a success in my opinion.